Good morning. Welcome to the Medicare Advisory Panel on Clinical Diagnostic Laboratory Tests. My name is Sarah Harding. I'm a member of the Medicare Clinical Laboratory Fee Schedule team, which is in the Division of Ambulatory Services. I'm also standing in today as the designated federal officer for the CDLT panel. I want to begin by just saying how excited we are to be getting back to somewhat of an in-person meeting with this. Those of you who have been involved with us in the past several years know that this has been online entirely, I think for the past 5 years. So we are definitely excited to see some of you in person, but don't worry if you're online, you're also just as involved, you're not missing anything. Please also welcome the members of our advisory panel. We have 3 in the room, which is fantastic, and then the rest are online. This meeting is an opportunity for the CDLT panel to provide input and recommendations to the Secretary and Administrator of CMS on the appropriate basis for establishing payment amounts for new or substantially revised HCPCS codes being considered for Medicare payment under the CLFS for next calendar year. This meeting is also a forum for the public to respond to direct questions posed by the CDLT panel members in a real-time manner. We will only be discussing codes addressed in the CLFS 2026 test code update file, which is found on our CLFS Annual Lab Meeting webpage. Comments and recommendations on whether codes identified, so comments and recommendations on codes that are currently in gapfill that were discussed last year are not a part of this meeting. That is a separate process. Consistent with the CLFS process, after consideration of the information from our public meeting held in June, those public comments, today's panel recommendations, and anything we get in the coming weeks, we will generally post preliminary payment determinations on our website, usually in early September of each year. So in other words, if you're new to this process, everything coming out of this meeting and the work that CMS will do subsequently will all be published online in the coming weeks. Again, usually it ends up being very late August or early September. After which we will have a public comment period on those preliminary determinations, accepting any comments. We welcome criticisms. We also love, hey, CMS, you got it right. This is perfect. And we accept them all. The final determinations after that public comment period are typically posted in late November. Usually, if we can, we get it out just before Thanksgiving. These are the finalized payment determinations for each of the codes that are going to be discussed today. Those go into effect January 1st, 2026. Reconsiderations. So after the final payment determinations, the public, you all have an additional 60 days from the time we publish those final determinations to request reconsideration of either the basis of payment, so whether we decided to gap fill or crosswalk, or the specific crosswalk determination for all of these codes that are discussed today. It should be noted that the requester of a reconsideration should be prepared to present at next year's annual laboratory meeting. If not, the code may be removed from the list of reconsidered codes. I think all of you probably have seen the updated agenda. That did occur this year for 2 codes. The requester did not show up at the public meeting, and so we have removed those codes from the agenda. So please just be advised of that process for next year. You will be able to comment on those reconsideration requests next year. But once a reconsidered code goes through this cycle again, any decision on it is final. Okay, a few housekeeping issues. The agenda and code list are found on our CMS website. For those on standby, any of the labs or associations, anything on the phone or in the room, if you are not speaking, please place your phone on mute. For input from standby speakers, we will seek input from standby speakers only when the CDLT panel has questions specifically for those stand-in speakers. So in other words, speakers cannot address the interim co-chairs or the panel members directly. They can only do so when called on for specific questions. We want to get through this agenda as quickly as possible, and we have umpteen opportunities for public comment following this meeting. So you will absolutely have your chance to fill in any gaps, ask any questions, make any comments to us. But we do ask that for this meeting, you expect to only speak when a question comes to you. Another important housekeeping rule is to simply honor the warnings when a discussion or a given speaker is potentially off topic. We will butt in and we will ask for that discussion to happen at another time. One other thing is we've done this before. If during the discussion of a specific code, a new recommendation is proposed and the panel needs additional time to review, the discussion for that specific code may be moved to the very end of the meeting so that the entire panel has a chance to review the new recommendation. We will keep a list of all the codes that need to be moved toward the end of the agenda and make sure that everybody knows that has moved. We hope to have a very efficient meeting. We look forward to interacting and hearing from the FACA panel as well as standby speakers. True housekeeping issues: bathrooms are down the hall. If you're new to this building, it's pretty simple. You pass them on your way in. The cafeteria is open once again, slightly limited in options, but it is open. There's also a small snack bar just outside the cafeteria. Cafeteria is back down this hallway, down the back staircase, and you'll basically see it from there. If you have any additional questions during the meeting, you can reach out to staff in the Division of Ambulatory Services, myself. We have staff over here who are helping out. If you have follow-up issues, you can submit all your inquiries to the panel resource box. The address is CDLT Panel at cms.hhs.gov. Before I turn this meeting over to the panel, I do just want to introduce management team of Division of Ambulatory Services. Sarah Shirey-Loso is over there in the corner. Michelle Cruz is even further in the corner. These are our directors and deputy directors. And that is it for me. So I am going to turn this over to our interim chairs, Dr. Leonards and Dr. Chong. And thank you very, very much for coming, and enjoy the day.
Chris, I think you're starting. You can introduce the panel. Okay, good morning, everyone. Okay. My name is Chris Chong, and I will serve as, and together with Dr. Joe Leonards, will serve as interim co-chairs during today's meeting. Again, welcome to the calendar year 2025 CDLT panel meeting, where we will seek information and input from our FACA panel. I would like to begin by performing a quick roll call of our panel members. So for each member, when you hear your name, please acknowledge that you're present. Vicky Baselski.
Present.
Pranu Chandra.
Present.
I'm present. Jillian Booker.
Present.
Megan Lansford.
Present.
Jennifer Jang. Jennifer Jang.
Can you hear me? Present. Yes. Okay, good. Thank you.
Okay, I see Dr. Joe Leonards.
I'm here.
Mark Rumpler.
Present.
Christine Smolzer.
Present.
Michelle Schoonmaker,
Present.
Heather Chappelle.
Present.
Now that we are done with roll call, I believe we have a quorum. I would also like to briefly note that in addition to the names of the panel members, you can see on the screen the source of the nomination for each panel member. At this time, I'd like to, Dr. Joe Lannis will brief the public on the advisory panel process for reviewing and categorizing the codes on the list of each year. Joe.
Thank you, Chris. Can you hear me?
Yes.
I hope so. Wonderful. So as stated by Dr. Chong, before getting started, I just wanted to briefly explain what the panel has done. So we've taken, a great number of codes on our list and we broke them down into categories. In general, we try to maintain consistency across the different categories of the AMA current procedural terminology uses as stated in their manuals. And then we look at the type of lab test, the specimen, and the analyte. So all of that goes into this decision-making process in terms of taking the large list of codes, for example, like genomic sequencing codes, as well as the microbiology and immunology codes, and we break them further down. With this breakdown, we look at the type of specimen, the analytes, the methodologies, the resources needed, as well as the indication of the test, just to name a few of the things that go into our consideration. And the whole purpose of this is that out of these major categories, we want to try to get recognition that within the AMA CPT code book, that to help to promote efficiency during the meeting and to help, of course, the panel members quickly begin to look at similar codes. You will notice that for today, we're going to cover many codes across multiple of these categories. You can follow the code list provided. I think you have this, if I recall correctly, it's the Appendix 3 of your agenda. And you can see some of the codes on the panel today here on screen right now. So we will begin, and there's a certain cadence to this that will be recurrent today. And the first code, which is a PLA code 0533U, has been reviewed by Heather, and I would like to hand that over. Let me just double check if that's correct. So the first step is that the subcommittee reviewer will introduce the code and describe the recommendation.
Thanks, Joe. 0533U is a new PLA code, a drug metabolism, genotyping, 16 genes. And there were no public recommendations or comments for crosswalk or gap fill for this code. The panel has identified 3 potential crosswalk codes that all have similar methodology, resources, analytes, and specimen type. The first code is 0516U, also drug metabolism, 40 genes. 0434U, another drug metabolism code, and then 0476U, a third drug metabolism code. So those are the 3 options in addition to the always present option of gap fill and then abstain for those panel members who may have a conflict of interest.
Wonderful. Thank you very much. I would like to ask the interim co-chairs if there are any new options for the panel to consider. Hearing none. Are there any questions or comments from the panel or from CMS? Also hearing none. If there are no additional comments or questions on this code 0533U, panel members, please cast your vote. So since it's this first vote, I hope that everyone has the link, and then I hope that the CMS team can provide the ballot.
This is Pranil Chandra. I'm not able to cast a vote because it shows that content is missing.
See the same thing.
You might have to refresh. I just had to do that as well. It's refresh in the browser. And then sign in.
Refresh, and then you may have to sign in again.
All right, I presume this will go faster for the second and third and all other codes.
We're still waiting on a few panel members. But yeah, this is our,
I was able to rectify things on my end. Thank you.
perfect.
Not able to sign in.
Okay. Who was that just spoke? I'm sorry.
Jerry Garner.
Okay, I'll send you an email. Sit tight.
Yeah, this is Chris Chong. I'm having trouble signing in as well.
So there should be a link in the chat for the panelists that will bring you to the voting website. I think you have to do a little registration with your name, but then you should be able to get in.
Okay, I got it. Thanks.
Please check that you did indeed select one of the answers. Even if you're abstaining, we need to have that too.
Sarah, how's it looking?
Almost there. Dr. Garner, I think it looks like you're still having trouble. Yes. Ah, there it is. Okay. We
Wonderful.
are ready to go. Thank you for your patience. That will never happen again, I promise. Smooth sailing from here on out.
I love that. All right, the next code is also PLA code, I believe for reconsideration 0423U. And I also hand it over to Dr. Chappelle.
So 0423U is code for reconsideration, another psychiatry/drug metabolism code. And there were two codes recommended by public comment. The first is 0345U, which is also a psychiatry/drug metabolism code. 0411U, again, similar type code. And then the panel has reviewed the recommended crosswalk codes and has looked at alternative codes with similar methodology, resources, analytes, and specimen types. And the panel has two additional recommended codes for crosswalk consideration, 0476U and 0516U. So those are the four crosswalk code options. And then, of course, the always present gap fill and abstain options.
Wonderful. Are there any new options for the panel to consider? Hearing none. Are there any questions or comments from the panel or from CMS?
One
There are no
question, this is
additional.
Michelle. Oh, sorry, Joe. This is Michelle Schumacher. Heather, did any of the recommended codes also report as a risk of toxicity? Did they include a risk assessment, or are they just evaluation of like Dukdale or individual targets?
Yeah, so based on the code description, which is really all we have to go by unless we're digging into the details of each test, it does not look like there is a risk of drug toxicity by condition in any of the other existing CPT codes that have been priced on the CDLT or on the CLFS. Sorry about that. So no, none of the codes specifically mention a risk of drug toxicity by condition.
Any other questions from the panel or from CMS? Hearing none. So if there are no other questions, then please cast your vote.
Okay, that's it. Thank you.
All right, it is smooth sailing. All right, the next code is 0552U. I'm handing it over to Heather.
This is a new PLA code for testing of genetic disorders in embryonic cells, reported as low risk or high risk for a familial genetic disorder in the pre-implantation setting. The recommended code for crosswalk by public comment was 81425. The panel reviewed this code and considered this code as well as other existing codes on the CLFS. And the panel's recommendation for another crosswalk or potential crosswalk code to 0552U is 0254U. This code is also a pre-implantation testing code that's looking at embryonic DNA and providing risk score or scores for other conditions in the embryo. And as always, gap fill and abstain are options as well.
Thank you, Heather. Any new options for the panel to consider? Hearing none. Any questions or comments from the panel or from CMS? All right. If there are no additional comments or questions, panel members, please cast your vote.
Okay, all the votes are in.
That's great. All right. Next code is 0553U. Handing it over to Heather.
So 0553U is another genomic sequence analysis test of embryonic cells in the pre-implantation setting. The existing code for crosswalk that was recommended by the public is 0254U. The panel looked at that code and searched for additional codes that may be appropriate for crosswalk. And the panel agrees with the public comment and recommended crosswalk of 0254U.
Thank you very much. Any new options for the panel to consider? Hearing none. Any questions or comments from the panel or from CMS? Hearing none. There are no additional comments. Panel members, please cast your vote.
All the votes are in.
Wonderful. The next code is 0554U. Again, handing it back to Heather, our subcommittee reviewer.
So 0554U is a new PLA code, again, a pre-implantation setting genomic sequencing analysis of embryonic cells. The public recommendation for a crosswalk code is 0254U. These codes are similar in all the things that are important in terms of methodology, resources, specimen type, analytes, et cetera. And so the panel has reviewed the recommended code and looked for additional codes that are priced on the clinical lab fee schedule. And we have recommended the crosswalk to 0254U, the publicly recommended crosswalk code.
Thank you. Any new options for the panel to consider? Any questions or comments from the panel or from CMS? Hearing none. If there are no additional comments or questions on this code, panel members, please cast your vote.
The votes are in.
Wonderful. The next code is 0555U, and I hand it over to Dr. Zhang.
Thank you, Joe. Can you hear me clearly?
Yes.
Okay, good. Thank you. I have a little problem with the microphone before. I just wanted to make sure. Okay, so I will be presenting the code 0555U. And as you can see, it's pretty similar to the previous code Heather has presented. Basically, it's another pre-implantation genetic assessment. So the public recommended the crosswalk to 0254U. Panel members has discussed among ourselves, and we do agree with the crosswalk to the 0254U. And gap fill will be second option, but we agree with the public. This should be crosswalk to the 0254U. Thank you.
Thanks a lot, Jennifer. Any new options for the panel to consider? Hearing none. Any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please cast your vote.
All the votes are in.
Thank you very much. The next code is a reconsideration code 81195, and I hand it over to Dr. Heather Chappell.
81195 is a cytogenomic genome-wide analysis in hematologic malignancies using optical genome mapping. The public recommended a crosswalk code 0331U, which is an oncology code using optical genome mapping to identify abnormalities. And so the panel reviewed the recommended crosswalk code and looked for additional codes that are already priced on the clinical lab fee schedule that may also be reasonable crosswalk codes, and the panel has agreed with the public comment and recommendation for 0331U as an appropriate crosswalk code.
Wonderful. Thank you, Heather. Any new options for the panel to consider? Any questions or comments from the panel or from CMS? Hearing none. So if there are no additional comments or questions on this code, panel members, please cast your vote.
All the votes are in.
Thank you. The next code is 8XXX4, and I hand it over back to Dr. Chappell.
So this code 8XXX4 is a new MOPATH Tier 1 code, cytogenomic genome-wide analysis using optical genome mapping. There was a public comment recommending for crosswalk a combination of codes. That is, they include 81229, 88262, 81404, 81243, 88275. So a combination of those codes, adding them together was the recommendation from the public for crosswalk. The panel reviewed this code, reviewed this crosswalk recommendation and also looked for alternative codes already priced on the clinical lab fee schedule that would also be an appropriate crosswalk for this code. And the panel recommends 0260U as an appropriate crosswalk for 8XXX4.
Thank you. So there's a total of 4 options here. Any new options for the panel to consider? Hearing none, any questions or comments from the panel or from CMS to 8XXX4? Also hearing none, if there are no additional questions, then panel members, please cast your vote.
All votes are in.
Wonderful. The next is a PLA code in the transplantation setting X159U, and I hand it back to Heather Chappelle.
So X159U is a new PLA code for transplantation using mitochondrial or microRNA and reporting an algorithm of risk for liver allograft rejection. There was a publicly recommended crosswalk for this new code. That crosswalk recommendation is 0018U, which is an oncology microRNA test, also reporting out risk of malignancy in this case. The panel reviewed that recommended crosswalk code and searched the clinical lab fee schedule for alternative codes and tests that were already priced. And the panel has identified 2 additional codes for consideration, 0534U, which is an oncology microRNA test looking at 32 variants and reported as a risk score. And then 0069U, also an oncology microRNA test reporting with an algorithm reported as an expression score. So there are 3 crosswalk code options and then the always present gap fill and abstain options for this code.
Thank you. Any new options for the panel to consider? Hearing none. Any questions or comments from the panel or from CMS?
This is
We
Michelle. I
may
just
need
have
to
one quick
revise,
question.
we may need to revise the ballot to also include 0534U.
Yeah, the ballot's just missing one of the crosswalk codes.
Dr. Chandra, you said 054U? What was
I
the code?
believe-
0534U.
0534U. Yep.
Correct.
Any additional comments from the panel or from CMS?
Hi, this is Michelle Skumaker. I had a question. Was there a multiplier on the 0018U recommendation by any chance?
I feel like I remember seeing that there was a modifier at some point and then I feel like it got lost along the way.
Because in the public recommendation, there's a note saying that the reduction is based on a reduction in the number of genes from 10 to 4.
I think there was a there was 0.85.
Yeah.
Okay, so let's pause just for a second. So we did actually remove 0534U as an option because it is a new code this year. So we took that off as an eligible code. So we're actually going to re-edit the poll to take that off so that we don't have to redo this. And now let's address the multiplier. Is that something the panel would like to officially recommend as an additional option or as an edited option?
I'm just wondering if that was the original recommendation. If you look at the notes from the public, there's the last line is there's a reduction based on the reduction in the number of genes from 10 to 4.
Yes, I do see that.
And even above that, Michelle, just right after crosswalk or gap fill recommendation, it says crosswalk to 0.85 of code 0018U. So that was in there early on and and got lost. So that was actually the public recommendation is 0018U times 0.85.
0.85. Yes, ma'am. Okay, we will make that addition.
And
Your
then just
ballots
to-
should.
Yep.
1 second. Just let it update and you may have to refresh your screen. We're having connection issues over here.
It's a good point about the 0534U because that's actually the next code, so that cannot be crosswalked to.
Okay, just stand by for a slight technical difficulty here. Okay. It may be because, you know what, go back, cancel it. And clear responses. Clear. Now try it.
Do we maybe move this to the end?
Sure, if you want to move on, we can move on. We'll take care of it and come back to it. Thank you.
All right, the next code is the one that we just wanted to crosswalk to, which is 0534U. And let me just check. I don't
I think
see.
that one's mine.
Yeah. Okay.
This is Jillian Hooker.
Jillian, yes.
That
Yes, I see.
code 0534U is a new PLA code analyzing 32 different variants and reporting out as an algorithmic risk score for prostate cancer. The public recommendation was to crosswalk to 0018U, and the panel, after reviewing the methodology, contents, and reporting of this test, agrees with that recommendation. So the ballot is 0018U or gap fill, or obviously abstain.
Thank you, Gillian. Any new options for the panel to consider? Hearing none. Any other questions from the panel or from CMS? Hearing none, then panel members, please cast your vote.
Votes are in.
Wonderful. And now I want to hand it over for the next code to Dr. Chris Chong.
Okay, this I'll hand it over to Vicky or Michelle to present 0531U.
Thank you, Dr. Chong. This is Michelle Skimmaker. I'm going to take the first block of these infectious disease codes. Code number 0531U is an infectious disease test identifying 673 organisms that are acid-fast bacteria, invasive fungi by NGS on plasma. The public recommended several codes which the committee reviewed, and we didn't think that any of them really fit. 0326U and 81520 were codes that were performed on FFPE tissue and reported a risk score or algorithm, not individual discrete identification. And the 048U and 0152U, while both using NGS, they targeted all types of pathogens such as all bacteria, viruses, and parasites, in addition to acid-fast bacteria and invasive fungi. For that reason, the subcommittee recommended that this could be gap-filled.
Thank you. Are there any new options on the panel to consider? Hearing none, are there questions or comments from the panel or from CMS? Hearing none. If there are no additional comments or questions on this code, panel members, please now cast your vote.
Votes are in.
Next, X177U.
X177U is another infectious disease test. It's a DNA evaluation of 44 organisms, including bacteria and fungi, on urine using next-gen sequencing. The public recommended a crosswalk to 0504U, which is a PCR assay of 17 pathogenic organisms. The subcommittee, because this is a next-gen sequencing test with 44 discrete results, we recommended that the code be gap-filled.
Thank you. Are there any new options from the panel to consider? Hearing none, are there any questions or comments from the panel or from CMS? Hearing none, if there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Next code, 871XX.
871XX is a new CPT code for susceptibility studies of carbapenemase enzymes with 3 different enzymes. The public recommendation for this was gap-fill, and we actually found CPT code 87185, which is susceptibility studies using enzyme detection per enzyme. We feel this is a better crosswalk given the similar purpose and analytes, and it's done on culture isolates. We also recommend a multiplier ×3 because you're evaluating 3 enzymes. So the final recommendation would be 87185 ×3.
Okay, thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Can we move on to the next? Okay, 8XXXX. Dr. Shoemaker.
Yes, so 8XXXX is infectious disease antigen detection with direct optical observation for SARS-CoV-2 and also influenza types A and B reported separately. The public recommendation was 87811 plus 87840. However, after discussion, typically there would be a multiplier ×2 if influenza was reported, type A and type B were reported independently. And so the subcommittee recommended a crosswalk to 87811 plus 87804 ×2.
Thank you. Are there any new options on the panel to consider? Are there any questions or comments on the panel or from CMS? Okay. If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Next code, X166U.
X166U was a new code for Borrelia burgdorferi, antibody detection of 24 recombinant protein groups by immunoassay for IgG. I don't believe we received any public recommendations on that code. And so because the immunoassay method is not clear in the descriptor or how the 24 protein groups were reported out, the subcommittee recommended gap fill.
Thank you. Are there any new options on the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Okay, thank you. Next code, X182U.
X182U is an infectious disease code for the semi-quantitative measurement of pancreatic stone protein concentration in whole blood reported as a risk of sepsis. Again, we did not receive any public recommendations for this code. And so the subcommittee recommended gap fill on the basis that we could not find any comparable analyte or method.
Thank you. Are there any new options for the panel to consider? Are there any questions or comments on the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Thank you. Next code is 0574U.
For 0574U, this culture filtrate protein for a 10 kilodalton testing in serum or plasma for tuberculosis. Again, we did not receive any public recommendations for this code, and the subcommittee recommended gap fill as there's no comparable code matching the analyte type, and we are really unclear about the liquid chromatography or mass spec methodology that was used.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Thank you. Next code is 0556U.
0556U, the testing of pathogen-specific DNA and RNA by real-time PCR for 12 targets from nasal or throat swab reported as detected or not detected. We did not receive a public comment on this code, but we recommended a crosswalk to 87633.
Thank you. Are there any new options for the panel to consider? Are there any questions or comments on the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Next code is 0557U.
0557U is a code for testing bacterial DNA markers in vaginal fluid reported as detected or not detected for bacterial vaginosis and vaginitis. The subcommittee identified 81514 as a comparable CPT code based on the fact that it's a similar specimen, purpose, method, and resources in analyte reporting. The recommendation is a crosswalk to 81514.
Thank you. Are there new options for the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Thank you. Next code is 0563U.
0563U, pathogen-specific nucleic acid, DNA, or RNA testing for 11 viral and 4 bacterial targets and upper respiratory specimens reported as positive and negative for each organism. There was several codes recommended. One was 0115U and also 87633 based on the number of targets. When we did some research, we noticed, or we saw that 8155U was crosswalked to 87633 in the past. So we recommended a crosswalk to 87633 based on the similar specimen type and method and number of analytes and resources. So the recommendation is crosswalk to 87633.
Thank you. Are there any new options for the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Thank you. Next code is 0564U.
0564U is similar to the previous code as pathogen-specific DNA or RNA analysis for 10 viral targets and 4 bacterial targets in an upper respiratory specimen, each pathogen reported separately. Again, the subcommittee recommended a crosswalk to 87633 based on the similar specimen type, methodology, number of analytes, and resources.
Thank you. Are there any new options on the panel to consider? Are there any questions or comments on the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Okay. Is 87626 now removed from consideration? Because I know this is all struck off now. Okay. Next, we'll go on to X171U.
This is Vicki Basowski. I'm going to take over here now with the remaining infectious disease codes. This is a molecular test for infectious disease prediction. It looks at a number of host genes to determine what the likelihood of a bacterial or viral infection is. There was a public comment with the recommended crosswalk to 0203U, which the subcommittee agreed with based on the number of targets, the method, algorithmic reporting requiring similar resources.
Thank you, Dr. Basowski. Are there any new options from the panel to consider? Are there any questions or comments from the panel from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
The next code is X183U. Dr. Basowski.
X183U is a PLA code for detection of a number of tropical fever pathogens. As described in the descriptor, it's a total of 9 targets. I believe there were public recommendations for crosswalk to another multiplex code with a similar number of analytes, 87506, and we agreed with that crosswalk based on similar method, number of analytes, method, and resources required.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments on the panel from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Let me scroll. I think Dr. Lannis, you take over from here.
Thank you, Chris. The next code is 0536U, and I hand it over to Dr. Zhang.
Thank you, Leonard. Okay, so for the code 0536U, I believe there are public recommendations, including 3 codes, 0453U, 0488U, and 0494U. We believe we have the panel discussion. We believe the 0453U appear to be the most appropriate test code to crosswalk to because of the similar methodology as well as targeted genes and as well as the reporting method. The other 2 are using 0488U and 0494U are used different methodology. Therefore, we believe the 0453U is the most appropriate code to crosswalk to. Thank you.
Thank you, Jennifer. Any new options for the panel to consider? Any questions or comments from the panel or from CMS on 0536U? Hearing none, then panel members, please cast your vote.
All votes are in.
Wonderful. The next code is 0523U. Handing it back to Dr. Zhang.
Thank you. So for code 0523U, this is for the oncology test code with the DNA qualitative next generation sequencing test utilizing the formalin fixed paraffin embedded tissue. So there is a public recommendation for code 0022U minus 0144U. As you can see, 0022U covers both the DNA and RNA. That's why they recommend to minus 81449, which is for the RNA testing. However, our panel, our committee has researched the code. We believe 81445 is the most appropriate. It's similar methodology, integrate DNA analysis as well as with the similar genes as well. So panel, subcommittee panel recommended 81445 for this test code.
Wonderful, thank you.
Mhm.
Are there any new options for the panel to consider? Hearing none. Any questions or comments from the panel or from CMS? If there are no additional questions, panel members, please cast your vote.
All votes are in.
Wonderful. The next code is 0538U. And handing it back to Dr. Jean.
Thank you. So for code 0538U, another oncology NGS analysis, use similar formalin fixed paraffin embedded tumor tissue for DNA analysis. Now, actually, I think there is a missing right here. I'll need to add on the one more code. Joe,
Yeah,
can we do that? I need on the number 4 choice should be the 81459.
That's correct.
Mhm. Because both 81459 and 0334U, which is recommended by the public, have a similar methodology, similar specimen, and a similar analysis. However, the only difference is for 0334U specified number, gene numbers 84 or more, 81459 does not.
So Dr. Jiang,
Mhm.
the display document on your screen does not
Yeah.
have that option, but your
That's correct.
voting ballot
Oh,
does.
excellent. I'm glad to hear that. Thank you for the clarification. So, yeah,
I apologize for the miscue, but yes.
it's okay. No problem. Thank you. Mhm.
Okay.
Okay.
Any other new options other than 81459 for the panel to consider?
For the panel, since we don't have the long descriptor up, although it is in the next code.
Yeah, it is in the next code. That's correct.
Okay, so it's right there if you need
Yeah.
to read the long descriptor for 81459. Thank you.
All right. If there are no other questions from CMS or the panel, please, panel members, cast your vote.
All the votes are in.
The next code is 0543U. Again, Dr. Jean.
Thank you. It's a similar code as you seen at above. Again, another oncology for solid tumor NGS analysis for DNA gene mutations and using the FFPE tissue. So basically, I already talked about the 81459 in earlier code, and another code was recommended by the public is 0379U. Again, the difference is 0379U did specify the gene code. 81459 does not. But the panel think 81459 is the most appropriate because simplification for all. As well, everything's similar.
Wonderful.
Thank you.
Any other new options for the panel to consider? Hearing none. Any questions or comments from the panel or from CMS? Hearing none. Panel members, please cast your vote.
All votes are in.
Perfect. The next code is X164U, which is an oncology RNA-based test for the indication of cutaneous melanoma. You see a qPCR of 10 genes. The subcommittee recommendation is a crosswalk to 81525 because of similar complexity, effort, and overall resources used. And an additional consideration was 81529, which the panel considered to be not as closely matched due to the different in content and slightly different amount of analysis needed. So the subcommittee recommendation for X164U is a crosswalk to 81525. And I open it up for additional comments or new codes that the panel should consider. Hearing none. Any questions or comments from the panel or from CMS? Hearing none either. So panel members, please cast your vote.
All the votes are in.
Thank you very much. The next code is X172U. And here, this is an mRNA-based gene expression profiling assay with 216 genes. The subcommittee reviewed the content and methodology and identified 81456 as the most appropriate crosswalk. Let me just see. There's a note. No, I think that this was the main alternative. Okay. Any new options for the panel to consider? Any questions from the panel or from CMS? Hearing none. Panel members, please cast your vote.
All votes are in.
Thank you very much. The next code is X185U. Handing it back to Jennifer.
Thank you, Joe. So for this code, X172U, and there is oncology test based on the extracted mRNA profiling test. However, we did not have any public recommendation, and we did not have any particular information of the company either. I think I'm sorry, I want to correct myself. Are we talking about the number 31, X185U? Yeah,
correct.
31, right, okay, all right. I'm sorry, I apologize. X185U is the code I'm discussing right now. This is a code that not only integrate RNA expression, but also they also perform multiplex immunofluorescence for 20 proteins expression as well. This is a difficult one because panel had the discussion and we find 81521 seems the most appropriate code, however, does not including the test for the multiplex immunofluorescence for integrating for the 20 protein expression. Therefore, panel decide gap fill is probably the most appropriate code, the choice for this test code. 81521 is something to think about, however, not the most appropriate one for this test code at this point. Thank you.
All right. Are there any new options for code X185U? That's in order number 31. Any new codes to consider? Any questions or comments from the panel or from CMS? Right, panel members, please cast your vote.
All the votes are in.
I hand it back over to Dr. Chong.
Thank you. 0527U.
This is Vicki Baselski. We're back to some infectious disease tests here. 0527U is a nucleic acid amplification test to detect 3 analytes, HSV1, HSV2, and varicella zoster virus. We recommended a crosswalk to 87631, which is a different sample type, but has a similar number of targets, methodology, and analytes. There was also a recommendation, public recommendation for a crosswalk to 0455U. In researching this, that code had previously been crosswalked to 87631. So for consistency, we felt like that was the more appropriate crosswalk, 87631.
Thank you, Dr. Pasowski. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Can we scroll to the next code? 0528U.
0528U is another nucleic acid amplification test for lower respiratory tract infectious agent and antimicrobial resistant marker detection, 18 bacteria, 8 viruses, 7 resistance markers for a total of 33 targets. There were a number of recommendations for looking at additive codes 87633, 87632 to get to those 33 targets. However, in looking at the existing codes, we felt like 0321U, although a different sample type, did cover the number of targets using a similar method and similar resources. So our recommendation was to crosswalk to 0321U.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? There are no additional comments or questions on this code. Panel members, please now cast your vote.
All votes are in.
Okay, next code 871X1.
871X1 is a susceptibility study test using nucleic acid amplification on isolates, looking for carbapenem resistance genes. A total of 5 genes are amplified in this test. Although a recommendation was made by the public to crosswalk to a direct specimen test that had a similar number of analytes, 87505, we felt like because this was a test done on an isolate, that a culture typing code was more appropriate. Thus, we proposed 87150 times 2. This is culture typing on isolates by an amplified probe, and it would be used for each probe. So again, the recommendation by the subcommittee is 87150 times 5.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Okay, next code is 87513.
87513 is a code that is reconsidered from last year. It's infectious agent detection by nucleic acid for Helicobacter and for clarithromycin resistance in Helicobacter. I believe last year we were unclear as to whether this was done on a direct specimen or on an isolate. So the proposal assumed it was done on an isolate. However, in this year's presentations, it was clear that this is done on a specimen. I believe it's a fecal sample. So the code 87564, which is another code done on a direct specimen that identifies M. tuberculosis and rifampin resistance, was a more appropriate crosswalk. So we are recommending crosswalk to 87564.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Next code is 8XXX9.
8XXX9 as another nucleic acid amplification test, multiplex test for joint space pathogens and drug resistance genes. It looks for 26 or more targets. The recommendation, I believe a public recommendation for a crosswalk to 0505U was based on there being a comparable number of analytes in that assay, although the specimen type is different. The recommendation of the subcommittee was similar to that of the public 0505U based on the number of analytes. So again, although the specimen is different, it's similar method, similar number of reportable analytes, and therefore resources required.
Thank you. Are there any new options for the panel to consider? Are there any questions or comments from the panel or from CMS? There are no additional comments or questions on this code. Panel members, please now cast your vote.
All votes are in.
Okay, thank you. Next code is 8XXX2.
8XXX2 is again a multiplex nucleic acid amplification for 2 analytes only, chlamydia trachomatis and Neisseria gonorrhea. There was a public recommendation to crosswalk to 0402U times 0.5, I believe, because that is a test for actually 4 different genitourinary pathogens. However, based on coding convention and other similar tests that detect only these 2 agents, we believe the more appropriate crosswalk is the agent-specific crosswalk to 87491 plus 87591.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Next code is X180U.
X180U is a PLA code for detection of genitourinary pathogens and associated antimicrobial resistance genes. There are a total of 46 targets that are identified in this test. We believe that a crosswalk to 0321U for GU pathogens with a similar number of analytes is appropriate. Similar method, similar analyte, similar specimen type.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
G 05.
E0567, your final infectious disease assay for this session is for infectious agent detection for binucleic acid amplification for hepatitis C screening technique. This was presented at the public meeting as a point of care test. However, it is an analyte specific methodology, and we believe that a crosswalk to an analyte specific identical analyte 87521 without a multiplier is appropriate. The public recommendation was for a multiplier to account for the point of care methodology, but we felt like it was a straightforward analyte specific crosswalk to 87521.
Thank you. Are there any new options on the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Dr. Lennons.
Thank you. The next code is 0540U, and I'm handing it, or actually I'm managing this one, sorry. This is a transplant medicine code, a quantification of donor cell-derived cell-free DNA. Just for the panel, there was also a prior option that was wrong. The correct crosswalk recommendation by the subcommittee is 0493U due to similar effort, amount, and scope of assay. So the subcommittee's panel recommendation is 0493U as a crosswalk. I open it up for new options for the panel to consider. Hearing none. Any questions from the panel or from CMS? Panel members, please cast your vote.
All votes are in.
Perfect. The next code is 0530U. And this is Dr. Chandra.
Thank you, Joe, and good morning to everyone. This is a CPT code we evaluated. It's an oncology pan-tumor ctDNA code performed on plasma utilizing NGS that assesses for microsatellite instability, TMB, along with an interpretative report. I believe there was a recommendation to crosswalk to 81455. We did look at this internally, and we wanted to just be consistent with the resources and the fact that this is a plasma-based assay. So we actually would recommend a crosswalk to 81464, also noting the public recommendation of 81455.
Thank you, Dr. Chandra. Any new options to consider? Hearing none. Any questions or comments from the panel on code 0530U? Hearing none, then panel members, please cast your vote.
All votes are in.
Thank you. Next is 0539U, Dr. Chandra.
This is an oncology cell-free DNA code as well of 152 genes, looking at SNVs, indels, gene rearrangements, copy number alterations, MSI using whole blood samples. There was a public recommendation of 0326U. Again, we wanted to be consistent with historical recommendations and also with the resources required. The 0326U is a cell-free DNA that's undergone the FDA review process. And so we recommend an 81464, which we feel is more appropriate to the test as well as the resources required.
Wonderful. Any new options for the panel to consider? Any questions or comments from the panel or from CMS? Hearing none. Panel members, please cast your vote.
All votes are in.
Wonderful. Thank you so much. The next code on the list, number 43, is 0560U. This is a whole blood and a tumor tissue based baseline assessment for the design and the construction of a personalized variant panel. And there were several considerations. The best fitting one is 0306U for consistency as well as complexity. It is also cell-free, and it's a panel for future comparison to evaluate for minimal residual disease. So the subcommittee recommendation is crosswalk to 0306U. Any new options for the panel to consider? Hearing none, any questions or comments from the panel or from CMS? Hearing none, then panel members, please cast your vote.
All votes are in.
Wonderful. The next code is 0561U. Again, genome sequence analysis and cell-free DNA from whole blood, subsequent assessment for comparison to the initial assessment. The subcommittee reviewed this and identified 0307U, similar to the subsequent code of the last crosswalk, as a patient-specific panel, cell-free DNA for subsequent assessment. Again, it seems to be matched almost perfectly. The subcommittee recommendation is therefore crosswalk to the option 3 as portrayed on the screen here. I open it up for new options for the panel to consider. Hearing none. Any questions or comments from the panel or from CMS? All right, then panel members, please cast your vote.
All votes are in.
Thank you. The next code is 0562U. This is a target genomic sequence analysis, 33 genes, SNVs in plasma, and reported as the presence of actionable variants. The subcommittee reviewed this and identified the PLA code 0388U as the closest match due to the description, the similar scope, as well as reporting, as well as the effort. So that's the subcommittee recommendation. Any new options for the panel to consider? Hearing none. Any questions from the panel or from CMS? Right, panel, please cast your vote. Do we have all votes?
We're just waiting for one more panel member. Okay, perfect. All the votes are in.
Thank you. The next code, I hand it over to Dr. Chandra.
Thank you, Joe. This is a next generation sequencing CPT code assessing for DNA and RNA in 80 genes and 10 genes respectively in plasma. We acknowledge the public recommendation of 81455, and we looked at our historical recommendations as well as CPT codes from a plasma perspective, and we were not able to find a great match that also accounts for the RNA, MSI, and or TMB. So we agree with the public recommendation of the crosswalk to 81455 in this case.
Thank you very much. Any other options for the panel to consider? Any questions or comments from the panel or from CMS? Hearing none, then panel, please cast your votes.
All votes are in.
Perfect. The next code is X169U, number 47 on the list, targeted genomic sequence analysis, cell-free DNA from plasma with 521 genes, copy number assessment, gene rearrangement, MSI, including variant and actionability. The subcommittee reviewed this. Although the number of genes was not perfect, the amount of anticipated work, analysis, and scope of what's being reported seemed very similar. So the crosswalk recommendation by the subcommittee is to PLA code 0239U. I open it up for additional comments or other options that the panel should consider. Hearing none. Any questions or comments from the panel or from CMS? Hearing none, then panel members, please cast your vote. And I hand it back over to Dr. Chow.
All votes are in.
Okay, Dr. Garner, you can present 0524U.
Yes, thank you. This is a new PLA code. It is an immunoassay ratio panel for preeclampsia. The public recommendation was for a crosswalk to 0482U. The subcommittee reviewed the components and agreed these are the same components and methodology. Therefore, it is our recommendation to crosswalk to 0482U.
Thank you. Are there any new options on the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Okay, next code is 0550U, Dr. Garner.
Yes, thank you. This is an ELISA panel for the risk of high-grade prostate cancer. There was no presentation or public recommendation received, and therefore, in research in the subcommittee, the recommendation was to crosswalk to 81539 divided by 2, or stated differently, 81539 times 0.5.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Thank you. Next code is 0558U. Dr.
Yes, this is a new PLA code. It's an ELISA biomarker test for colorectal cancer. There was no public presentation submitted or recommendation received. The subcommittee reviewed the information available and recommended a crosswalk to 86316.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Okay, Dr. Gardner, next code.
Yes, thank you. ELISA biomarker test for a breast cancer marker. It is again, a no presentation submitted, no public recommendation received. Subcommittee reviewed what we could find and recommended a crosswalk to 86316.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments on the panel from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Thank you. 0573U.
Yes, this is a new PLA code. It's an oncology panel linked to pancreatic cancer. We did receive a public recommendation, which included a crosswalk to 82948 plus 82378 plus 2/3 of 0080U. Upon researching the submitted information, we determined that it would be better to gap fill this particular code because the rationale is that Raman spectroscopy doesn't align well with the recommended crosswalk.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Can we scroll? Okay. Next code is X178U.
Yes, this is a new code for a panel of 3 biomarkers for prostate cancer detection. And in looking at the recommended crosswalk to 81539, the subcommittee recommendation actually aligns with that crosswalk to 81539.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Next is X187U.
Yes, this is an immunoassay panel for pancreatic cancer. The recommendation from the public was to crosswalk to 0342U. Upon review, the subcommittee came back with a recommendation to crosswalk to 81539.
Are there any new options on the panel to consider? Are there any questions or comments on the panel from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Next code is X162U, Dr. Rumpler.
Thank you, Dr. Chong. X162U is an application in oncology. It's a serum analysis of 39 glycoproteins by LC tandem mass spectrometry, and it's reported as a likelihood for malignancy. There was limited information available in the public meeting. We also do not have a public recommendation for this code, but the subcommittee is recommending a crosswalk to 81503. And 81503 specifically covers proteomic assays analyzing 7 or more proteins with a software algorithm that also generates a malignancy risk score, which closely matches the ovarian cancer's tests methodology.
Thank you. Are there any new options on the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
Votes are in.
Thank you. I'll hand it over to Dr. Leonards.
Thank you very much. The next code is 0549U, which is an oncology urothelial cancer quantitative methylation real-time PCR. There are certain gene targets, and upon review, the subcommittee reviewed and found that PLA code 0012M, which is also an oncology, also urothelial, here RNA expression marker, but PCR for 5 genes would be the closest similar. There was also another consideration, which is 0229U, which is a BCAT1 and IKZF1 promoter methylation analysis, but the subcommittee felt that by number of genes and complexity, while being a methylation assay, that the code 0012M would be the closer match. So that's the subcommittee recommendation. Are there any new options for the panel to consider? Hearing none. Any questions or comments from the panel or from CMS? Hearing none. Then panel members, please cast your vote.
All votes are in.
Thank you very much. The next assay, 0565U, is a very interesting next-gen sequencing methylation pattern assay detecting over 6,000 epigenetics alterations in cell-free DNA. An algorithm, reported as cancer signals. The subcommittee discussed this, took many different codes into account, but did not feel that any code is closely similar. There were a few that were roughly 2,500 genes. The methylation region was determined, but ultimately the subcommittee felt that the gap fill process would probably be the closest to match this. So I'm opening it up. Any new options for the panel to consider for code 0565U? Hearing none. Any questions or comments from the panel or from CMS? Hearing none, then panel members, please cast your vote.
All votes are in.
Thank you very much. The next code is 0566U. This is a qPCR-based analysis of 13 differentially methylated regions in pleural fluid, and an algorithm reported as qualitative results for against lung cancer. The subcommittee reviewed this code and the description by complexity and identified that there's one potential crosswalk, which is 0398U. However, while the committee was split, the no perfect match and therefore recommendation for gap fill was the majority of our folks on the committee. And I open it up in case there are any new options for the panel to consider. Hearing none, any comments or questions from the panel or from CMS? Hearing none, then panel members, please cast your vote.
All votes are in.
Perfect. The next one is, let me just double check, 0569U, if I'm not mistaken. This is a methylation markers in self-recirculating tumor DNA in whole blood. Again, algorithm detection as presence or absence of ctDNA in solid tumors. The subcommittee had several options here for consideration. We identified 0493U as a transplantation based assay. This is donor-derived cell-free DNA. It's definitely not a perfect match. And there was an 0537U, which is an oncology-based assay. But in this case, the descriptions didn't match perfectly. So the subcommittee recommendation was ultimately, while offering the other PLA codes as potential crosswalk options, the recommendation was for gap fill. So 0569U recommendation for gap fill, and any other options for the panel to consider. Hearing none. Any other questions or comments for the panel or for CMS? From CMS. Hearing none, then panel members, please cast your vote.
All the votes are in.
Okay. The next code is 0572U, which is a high throughput telomere length quantification assay. This is done by fluorescent in situ hybridization testing in whole blood. And there's an algorithm as a report for the risk of prostate cancer. There was a few options that looked similar. One was 0537U, again, a cell-free assay, epigenomic pattern rather than telomeric length. Not sure if both are high throughput, so there's definitely some discrepancies, but that was the closest. So the subcommittee felt strongly to offer this crosswalk option. However, as a recommendation, we voted for gap fill. So 0572U gap fill recommendation by the subcommittee, and I open it up for any new options or considerations for the panel. Hearing none. Any questions or comments from the panel or from CMS? Also hearing none, then panel members, please cast your vote.
All votes are in.
All right. Then I see the next code, which was 0464U. This was for reconsideration, but is removed, correct?
Correct. Correct.
Just checking. Wonderful. Then the next one is 0420U, which is an urology mRNA expression profiling real-time PCR assay for the genes portrayed here on screen. It's a combination, an analysis of 6 single nucleotide polymorphisms, as well as TERT and FGFR, as well as algorithm and a risk report. So we discussed this quite a bit, the crosswalk to 0012M plus a fractional crosswalk to, I believe, 0.35 times 0356U, which is another PLA code, was the recommendation. This is because of the combination of 2 different elements here that we felt would reflect this the best. So I believe this was a reconsideration code, and we also took into account, the other options were all not favored by the subcommittee. So here, option 3, portrait, where there's like a little line break, that's the combination recommendation by the subcommittee. I open it up for any additional new considerations or new options for the panel. Hearing none. Any questions or comments from the panel or from CMS? Also hearing none. Panel members, please cast your vote.
All the votes are in.
Thank you very much. The next code is 8XXXO. This is also mine. This is a central nervous system tumor DNA methylation analysis of at least 10,000 methylation sites. So very comprehensive assay. You see that there's also reported as the probability of matching a reference tumor family, class, and MGMT status or promoter methylation status if performed. The closest element or the closest similar description that the subcommittee identified was 0537U. However, different numbers, different purpose of the test. So the ultimate recommendation of the subcommittee was gap fill, but we left the additional PLA code in as an option. So the recommendation by the subcommittee is gap fill. Any new options for the panel to consider? Hearing none. Any questions or comments from the panel or from CMS? Hearing none, then panel members, please cast your vote.
All the votes are in.
Wonderful. This next code is X179U, an oncology panel, hematolymphoid DNA targeted genomic sequencing of 417 genes. Based on the complexity as well as the overall effort here, we looked at a crosswalk to 81455. And I open it up for new options to consider for the panel. Hearing none. Any additional questions or comments from the panel or from CMS? All right, hearing none, then panel members, please cast your vote. And I yield back to you, Chris.
All the votes are in.
Zero 0535U, Dr. Rumpler.
0535U is a code for perfluoroalkyl substances, otherwise known as PFAS, by liquid chromatography tandem mass spectrometry in plasma or serum. It's a quantitative method. There was limited information available on this in the no presentation, and there was no recommendation in the public meeting. So through our understanding, the subcommittee is recommending a crosswalk to 0457U, which closely aligns with the technology and the clinical purpose.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? Hearing none, panel members, please now cast your vote.
The votes are in.
Can we scroll? Next is 0541U.
0541U has an application in cardiovascular disease. It's again an LC tandem mass spectrometry quantitative measurement of 5 distinct HDL-bound apolipoproteins. The committee recommends a gap fill for this code because there's no existing CPT that fully represents the multiplexed LCMS analysis in addition to the algorithmic approach.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? Okay. Hearing no comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Next code is 0542U.
0542U has an application in renal transplant. This is a urine-based NMR spectroscopy measurement of 84 urinary metabolites it's a quantification of BK virus using a real-time PCR and serum creatinine. It's combined using an algorithm reported as a probability or risk score for allograft injury status. The subcommittee recommends a gap fill in this scenario because it combines an advanced multi-dimensional NMR method, which is unique to the CPT coding. So there's no other code similar to this. I believe there was a recommendation for a crosswalk of 81558, but the method itself is too unique and complex for a crosswalk at this time.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Next code is X161U.
X161U is another method for perfluoroalkyl substances, PFAS. It's 24 compounds via LC tandem mass spectrometry in serum. It's a quantitative method. The subcommittee recommends a crosswalk to 0457U because it closely aligns with the technology and the clinical intent And it also covers a panel of environmental toxins that uses LC tandem mass spectrometry.
Are there any new options on the panel to consider? Are there any questions or comments on the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Next code is X165U.
X165U has an application in nephrology specifically for diabetic chronic kidney disease. It's an enzyme-linked immunoabsorbent assay, or in other words, an ELISA for an apoprotein A4 and CD5L, and then it's combined with GFR rate. And then it also uses an algorithm to report a risk score for kidney function decline. It's a plasma-based method, and the subcommittee recommends a crosswalk to 0385U, which also uses a multiplex ELISA platform and with an algorithm risk score.
Thank you. Are there any new options on the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Okay, next code is X170U.
X170U has an application in therapeutic drug monitoring. It's for approximately 60 to 150 drugs and metabolites in urine, saliva. It's a quantitative method, and the methodology is liquid chromatography tandem mass spectrometry. It utilizes specimen validity and also an algorithmic analysis for a risk score for the presence or absence of the drug metabolite and for predicted adverse effects. There was also no presentation for this and no public recommendation. The committee recommends a crosswalk to 0328U because that code specifically describes a multiplex quantitative LC tandem mass spec assay for therapeutic drug monitoring of 120 or more drugs with an algorithmic analysis.
Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Okay, thank you. X181U.
X181U has an application in transplant medicine. It's an antibody non-HLA method that's utilized for blood. It's a flow cytometry method for a single antigen bead with 39 total targets and individual positive antibodies reported. The subcommittee recommends a crosswalk to 86833 because that also specifically covers antibody identification performed by flow cytometry, and it includes assays for HLA antibodies commonly used in transplant monitoring.
Thank you. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? There are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Code X186U.
X186U has an application in gastroenterology, specifically for irritable bowel syndrome. It's a measurement of IgG antibodies to 18 food items by microarray-based immunoassay utilizing whole blood or serum, and it's got a qualitative report. The subcommittee recommends a crosswalk to 0360U, and that's because this assay closely matches the technology analyte type and the clinical use. Both these assays use microarray to measure IgG antibodies for multiple food allergens.
Thank you. Are there any new options for the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Hand it back now to Dr. Lennox.
Thank you very much. Now we get to the fun part. All right, 0529U, which is a hematology assay, use case in venous thromboembolism with genome-wide single nucleotide polymorphisms, variant characterization, including factor 2 and factor 5 gene analysis. There was not a lot of information available. There was a consideration that there might be some similarity to certain other PLA codes, but one of the considerations was 81229, which was a cytogenomic assay. Definitely not ideal. And in lack of the more specific information about this particular assay, the subcommittee voted for gapfill, but leaving the option for 81229 as a possible crosswalk. Any new options? And I'm actually just seeing that, Gillian, this is your code. Did I capture this correctly? Sorry, I didn't mean to overstep.
Yep, you got it. We thought it was really similar to the 04466U code, which was gap filled. So we recommended gap fill.
Wonderful. So there was the 0466U is not an option yet, correct? Just to clarify.
Correct.
Wonderful.
Correct.
All right. Sorry for stealing your code. That won't happen again.
No worries. I thought maybe you saw a strategy to move through these things quickly.
No, absolutely not. Any additional questions or comments from the panel or from CMS? All right, panel members, please cast your votes.
All the votes are in.
All right, Gillian, back to you, 0532U.
Great. The next code is a new PLA code for rapid whole genome sequencing. The public comments had a recommendation to crosswalk to 0094U. The panel agreed with that recommendation and also recommends crosswalk to 0094U.
Wonderful. Any new options for the panel to consider? Any questions or comments from the panel or from CMS? All right, panel members, please cast your vote.
All the votes are in.
X168U.
Is for a rapid whole genome comparator sequence. There was some discussion on the subcommittee panel about whether adding a comparator for a rapid sequence proportionately increases the analysis time and work. And therefore it was recommended that we also consider crosswalk to the whole genome comparator 81426. So on the ballot are both 81426 as well as 0094U times 0.5.
Any other options to consider? Any questions or comments from the panel or CMS? If not, panel members, please cast your vote.
All the votes are in.
0567U.
0567U is a whole genome sequence analysis in which sequencing is done both with short read and long read sequencing, but reported out as a single report. Methylation of the genome is also reported. Public comment requested 2 times 0212U plus 0318U. In light of the fact that it's a single report, we also put on the ballot consideration of a single 0212U plus 0318U. And then also on the ballot is consideration of crosswalking to whole genome sequencing for proband 81425.
Okay, many options here. Any additional or new options for the panel to consider? Any questions or comments from the panel or from CMS? All right, hearing none, panel members, please cast your vote.
I think I didn't. I think correct me if I'm wrong, is 0212U already crosswalked to 81425? It's sort of the same.
No, there's 6 options on the screen, but only 5 on the vote. Is one of them.
I think option 3 and option 6 are essentially the same. Is that
Oh,
correct?
okay.
I think so.
Okay.
Yep.
Okay, all the votes are in.
Next code is X167U.
Yep, a rapid genome sequencing code. Public comment recommended we crosswalk to 0094U. The subcommittee met and agreed with that recommendation and is also recommending a crosswalk to 0094U.
Thank you. Any additional new options? Hearing none. Any questions or comments from the panel or from CMS for X167U? Hearing none, panel members, please cast your vote.
All the votes are in.
Okay, for 0544U, transplant monitoring following kidney transplant. We did not receive any recommendations in public comment for this code, but considered the code and thought that it was similar to 0493U and are recommending crosswalk to 0493U.
Thank you. Any new options for the panel to consider? Any questions or comments from the panel or from CMS? Hearing none, panel members, please cast your votes.
All the votes are in.
Thank you. X160U. Gillian, one more code for you.
Yes, is another transplant code, this time for liver rejection. In this case, public comment recommended a crosswalk to two codes, 81558 plus 0118U. However, in this case, there's a single risk score reported. And so we also recommended crosswalking to 81558 alone.
Okay, any other options? Hearing none. Any questions or comments from the panel or from CMS? Also hearing none, then panel members, please cast your vote.
Okay, all votes are in. I'm going to interrupt quickly, and we are moving so well, which is excellent. But I'm going to stop us for a lunch break. So we will reconvene, come back by about 1:20 so we can get all reconnected. For the panel, however, I have two notes for you. We are going to adjust the polls for that code early on, the X159U, to make sure the options are correct. And then a second code, 0565U, we did end up getting recommendations from the public on this. It was after the public meeting. They came in via email, and I think just slipped through our cracks. So I am going to send you guys the information that we got from them. They did send two possible recommendations. We're going to ask you to just look at it briefly, consider them, and we will have a revote after lunch. Those two codes that we are revisiting, we will do at the very end of the agenda for anybody who's listening. And I do not want to jinx anything, but I think the way we're moving says we are going to finish this up today, which is fantastic. So again, anybody on the phone, stay on the line because we will likely get to your code today. So any questions?
Yeah, please. The second code that you mentioned.
Yes, sir.
What was the code?
0565U.
Okay, perfect.
I will email the panel with the information. So please check your email over lunch, and then we will update the polls accordingly. And like I said, revisit it at the end of the agenda.
Yeah, that's very helpful because for the 0565U, we had nothing. And it would be very helpful to hear any or read any details over the lunch break. And the X159U, we're gonna do a vote at the end, correct?
Correct. Yeah, we just had a few alterations and the system didn't want to let us update it. So we'll do that when everything's quiet and revisit it at the end.
Perfect. Thank you.
Okay. All right, everybody have a great lunch. As I mentioned, cafeteria is downstairs, a little snack bar downstairs. We will see you back here at about 1:20 with a hard start at 1:30. Thank you. The end of our meeting. Okay, that's great that the recording is back on. We are going to start with number 80 in the order, which is X163U. As I mentioned before the break, we will tack on those two codes at the very end that their votes did not get finished, X159U and 0565U. So stay tuned for those. But I think unless there are any questions, problems, complaints, concerns, well, those you can keep to yourself. But anything else, please speak up. Otherwise, let's get going and finish this up. Number 80.
Okay. X163U, Dr. Garner.
Yes, so this is a neurology test for prion disease by quaking-induced conformational conversion using a CSF specimen type. The request from the public presentation is to crosswalk to 0035U. The subcommittee reviewed the tests and agrees with this crosswalk to 0035U based on the fact that they do use the same methodology.
Okay. Are there any new options for the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Okay, next code is 0570U.
Yes, this is a neurology test for traumatic brain injury, including glial fibrillary acidic protein and ubiquitin carboxyl terminal hydrolase L1. The request from the public presentation is to crosswalk to 0358U. The subcommittee reviewed these two tests, and while there is a difference in specimen type, the two tests do align on methodology and the number of components. So the subcommittee agreed to crosswalk to 0358U.
Okay, thank you. Are there any new options from the panel to consider? Are there questions or comments from the panel from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Next code is X184U.
X184U is a application in neurology for Alzheimer's disease. It's a plasma sample that looks at ApoE 2, 3, and 4 by LC tandem mass spectrometry, and it's reported as an ApoE prototype, excuse me. The recommendation was consistent. The subcommittee recommendation was consistent with the public recommendation of a crosswalk to X184U, which also utilizes a targeted LC tandem mass spec technique for peptide quantification for ApoE isoforms, which closely matches that technology.
Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS?
Hi, this is Michelle. Sorry, could you repeat what the subcommittee recommendation was?
So the subcommittee recommendation was a crosswalk to X184U. What you're going to find in the vote is two options. Excuse me, the subcommittee recommendation was 0412U. 184U is the code in question. The subcommittee recommendation is to crosswalk to 0412U. What you'll find is there are two options on the ballot, 0142U and 0142U times 0.33, the latter of which is the public recommendation.
Okay, are there any further comments?
But did that clarify, Michelle?
Yes, you threw me with this X184U recommendation.
Okay, if there are no additional
So
comments.
is I'm sorry, is there a reason that you think that it's the code without the multiplier?
The subcommittee is just giving a recommendation for the code. The multiplier is on the ballot because it was a recommendation by the public, but their rationale is that it is the difference in analytes that are measured between the comparator method and the method in question.
Are there additional comments or questions? Hearing none, panel members, please now cast your vote.
All votes are in.
Next code is 0521U, and the subcommittee is recommending 86200 times 4. This is 4 analyzed by immunoassay. There's no mention of an algorithm. So the crosswalk of 0312U, first of all, is not appropriate because it uses a combination of ELISA flow and indirect fluorescence, and there's an algorithm assessment. Whereas the long descriptor for the code submitted has no algorithm described. And so the subcommittee recommendation is 86200 times 4. Okay, are there any new options on the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Next code is 0522U. And there was a crosswalk considered 0472U, which has the same analyze and by same methodology that was submitted last year. But since there's a gap fill to be consistent, the subcommittee is recommending gap fill. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Next code is 0525U. And this one, although there were no materials submitted, this code is very similar to 0248U, which is testing by the same methodology. The only difference is 11 drugs versus 12 drugs. So the recommendation is to crosswalk it to 0248U. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
Okay, all the votes are in.
Next code is 0526U. For this code, there was no appropriate crosswalk when we examined other codes in the CLFS. And so we agree with the public submission of a gap fill? Are there any new options from the panel to consider? Are there any questions or comments from the panel from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Can we scroll to the next slide? Okay, code 0545U, acetylcholine receptor. The public has submitted a gap fill proposal. Various codes were examined, and 86363 is live cell based assay, but is by flow cytometry, so not not exactly the same. And so the subcommittee recommends gap fill. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All votes are in.
Thank you. 0546U is actually very similar to the code above. The submitter had requested a gap fill, and it's the same comment. While 86363 is a live cell based assay, it is by flow cytometry, and thus the subcommittee recommends a gap fill. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Thank you. 0547U, neurofilament light chain. The submitter requested a crosswalk to 0361U, and the subcommittee agrees with the agrees, and so the subcommittee recommends 0361U as is the recommendation. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Next code is 0548U, glial fibrillary acid protein. And the methodology used, immunoassay, is exactly the same as the previous code 0547U. And so the subcommittee recommends 0361U as the appropriate crosswalk. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
0551U, tau phosphorylated p-tau217. This is identical to 84393, but since that is in gap fill, to be consistent, the subcommittee is recommending gap fill. Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
All the votes are in.
Thank you. Next code, I think this is the last one, 0568U. This is for testing for ultra high sensitivity detection for Alzheimer's pathology of 5 analytes with algorithm. The submitter proposed 81503, and the subcommittee agrees that this is appropriate crosswalk and thus recommend 81503.
All votes are in.
Thank you. I think we'll hand it back to Sarah to take us back to the codes that were deferred to the end of the meeting.
Yes, sir. So let's go back to, in the meeting order, it's number 9. It's code X159U. Heather, I believe this was your code. All that we did for this, just as background, was to add the multiplier, the 0.85 that had been recommended by the public, and also removed the possible crosswalk of 534U because it is currently a new code. But I turn it to you if you want to represent or have any other discussion.
I'll just confirm that this is what it needed to be in the first place. We missed the modifier, and we also had 534 there by mistake. So these are the appropriate crosswalk codes. The 0018U times 0.85 was recommended by the public, and then the panel reviewed the code and recommended 0069U for crosswalk.
Are there any new options from the panel to consider? Are there any questions or comments from the panel or from CMS? If there are no additional comments or questions on this code, panel members, please now cast your vote.
So if there's no other discussion or other recommendations, I would ask you all to cast your vote now, please. Perfect, thank you. Okay, the next and last code for discussion today, going back to number 570565U. There are a couple notes on this one that I just want to make sure are presented. One is we did get a late comment that was after the public meeting that gave us 2 recommendations as possible crosswalks. One was, sorry, I'm just pulling up. One was 0410U. This is an oncology code for pancreatic, DNA, whole genome sequencing with 5 of that word, enrichment, whole blood or plasma, algorithm reported as cancer detected or not detected. It is up now on your, it is not up in there on your screen because I did not update that, but it is in your email if you need to read it again. The other possibility was 0507U. This is another oncology code. It's ovarian, DNA, whole genome sequencing with 5-hydroxymethylcytosine, enrichment, I could say it, using whole blood or plasma, algorithm reported as cancer detected or not detected. As additional information, the lab wrote to us. They said, this is a quote, "Please note that the next-gen sequencing does not only cover the 6626 epigenetic alterations, but our classifier uses those alterations to call HCC signal detected or not detected." The last change that needed to be made was there had been a very minor descriptor change. At the very end of that descriptor, it should not say in liver cancer. It should just say signal detected or not detected. That was the end of that. So I know that's a lot of information verbally. We can take some time and go over it. But the ballot that you see in front of you does reflect everything that is up for discussion. So please, any questions, comments?
Hi, this is Chris Chong. So what is the recommendation from the subcommittee?
So
In case
far,
it's relevant, is it also accurate that 0507U was previously crosswalked to 0410U?
yep.
Yeah, I mean, we can still consider both. They're both eligible as a crosswalk, but that's very useful to know that they're essentially, they were once called similar.
So the subcommittee has reviewed this, and there was nothing closely similar to the initial description. Now, those codes, both so I'm just pulling this up, 0410U and 0507U are whole genome, 5-hydroxymethyl cytosine enrichment. In other words, this is like, a methylation genome. And then there's either whole blood plasma algorithm and then a cancer detected yes or no in the 04 code for pancreas and the 05 code for ovarian. And if I understand the submitter, they're now claiming the same in hepatocellular carcinoma. So that's a fair crosswalk. The description, however, is not identical because, they use a specific number of epigenetic alterations, which I'm sure the other assays do as well. So that's, in terms of the effort, I think it's similar to, both of these. I can see that both are adequate crosswalks, depending on which you would consider closer, ovarian or pancreatic. But I think for that matter, both crosswalks are adequate. I don't know what the other panel members are thinking. One consideration is, of course, if you do whole genome methylation analysis, that's definitely much larger than 6,000 epigenetic alterations. That seems more like, but I would need to go back and check the long descriptors of those other two codes and then look at the manufacturer, how much they're actually analyzing of the whole genome. Do we have the submitter of 0565U on the call? So this is
I know they're on email. They've been emailing me to to our moderator, an individual named Laura Baden, Baden, from earlydx.com.
Laura, your line is unmuted.
Hi, can you guys hear me?
Yes, thank you.
Thank you so much for giving me a chance to explain our assay. We do whole genome next-gen sequencing on plasma, so for the cell-free DNA. And the only reason we listed the number of methylation sites that we look at is because those are the ones that our AI classifier looks at for that specific indication. So for different indications, the classifier may look at different sites depending again on the indication. But for HCC, cancer signal detected or not detected, it uses those. The wet work is exactly the same. We still extract the cell-free DNA. We still have to do the methylation. And then we still have to do the next-gen sequencing. We're using Illumina.
And you also do a 5-HMC enrichment?
Yes. So that's the methylation part. So yeah.
But just to verify, so whole genome is the same, plasma is the same, 5-HMC treatment is the same, and you report just a binary HCC present, yes or no?
Yes, cancer signal detected, yes or no.
So both the 041OU and the 0507U are identical, just using a different indication. So that both seem like an adequate crosswalk. It's just that the description doesn't give that away. So it's very helpful that your on the call and that you clarified that you also do whole genome sequencing, because that allows us to judge that the amount of effort in the wet lab is identical to those other crosswalk codes.
Yeah, I appreciate you letting me clarify.
No, of course. Any other questions from any of my other molecular or the broader panel members? All right. I hear none. Any questions from the panel or from CMS? Also hearing none. So then let's do a recast, and please, panel, cast your vote.
We are waiting for one more vote. Okay, we have everything. That is the end of our list. I'm not saying that we know there's no award for the most efficient panel meeting ever, but if there were, you can all raise your, no, thank you so much, everyone, for being here. The efficiency of this meeting goes entirely to the subcommittee and the amazing amount of work they did leading up to this meeting and being prepared with their recommendations. So thank you all to those online, to our co-chairs. Thank you very, very much for leading such an efficient and well-informed meeting. As I mentioned at the beginning of this, our next step is now for CMS to take over all of the information that we've seen through the public meeting, through this meeting, any other comments that we've gotten. We will be putting together a preliminary determination document that will be published online on cms.gov, likely at the end of August, early September. So please look for that. Any questions, you can find any of us at this table online, that CDLT inquiries email box, or CLFS inquiries, CDLT panel, all of those come to us. We are not a big team, so we see them all. But thank you again very, very much. Safe travels home, and we'll be in touch.